your final. I will have a test blueprint done today. So that's number one. Test blueprint. Lord. By tomorrow. Completed. And in your Jupyter grades. Okay? Are we clear? And that's going to tell you what's going to be covered on every question that's going to be on your final. This is for the final. Do you understand you still have a test next week? All right, so your test next week is just, let me get to that. Let me do this first. So test blueprint will be in your Jupyter grades tomorrow. Oh, it's not showing up? Sorry. So, could someone close the door? So the test blueprint is going to be sent to you via Jupyter Grades by tomorrow. So that'll break it down what exactly each question is going to cover. All right, it's all multiple choice. Your test is certainly going to be all multiple choice. How many questions is it going to be? There's two schools of thought, right? I'm going to share with you both schools of thought, and then I'm going to tell you what I've decided. All right. There's more than two schools of thought, but there's two that I've debated for a while. One school of thought is it's a 2-hour exam. You should have 99 questions, right? So it should be a lot of questions because it's a 2-hour exam. You don't want to give someone a 20-question exam, and then they have an hour and a half to sit around and cause trouble, right? That doesn't make sense. Plus, it's a lot of content. It's a whole semester. So how do I test a whole semester's material, right? And in less than 100 questions or so. I got two problems with that concept, that kind of strategy. Is it really testing what you know? If I'm giving you 100 questions, because can I really ask deep questions if you have to do a question a minute or something like that, a question of 2 minutes? It's multiple choice. You have a lot of reading. It's very stressful. You know what I'm saying? It increases a lot of issues. It adds on to the issues that come along with the final. And the other thing is that another thought that I have is that I don't have to ask you a question per vocabulary word, right? I don't have to ask you a question for every concept for me to understand if you understand the material or not. Literally the last test was, I think it was 30 questions. The last test was 30 questions. The membrane transport was about 30 questions, something like that. And I did a retest today during advisory. I just gave the child 4 questions, and it covered all the material on those tests. It was an essay question, but it was 4 essays, but 4 questions, and they finished it, and they proved that they didn't understand, and that's good. They kept the grade that they had. It was a retest, right? So the question is then, for you, how many questions do I have to ask for me to really understand if you understood what we've been talking about since August? And I feel like 30 questions is enough. I think I can ask 30 questions that, for instance, if I ask you a membrane question, why not ask you an osmosis diffusion question that includes the membrane, right? I don't have to ask you all kinds of questions about the membrane to understand if you understand what a membrane is. Does that make sense? So I feel I can do it in 30 questions. So I think that's what it's going to be. It's going to be a 30-question exam. And again, the blueprint you'll have by tomorrow. Then the next question is, you're not going to take 2 hours to do 30 questions. That's not going to happen. Yes, there's going to be little paragraphs and some analysis, remember? And I'll tell you, there's going to be reading graphs and graphing and experiment analysis and all that, right? Just like the other test. But it's still not going to take 2 hours. Max, I think it'll take you an hour to do the 30 questions, honestly. And that's at 2 minutes a question, right? Does that make sense? So I really think a lot of you will be done in half an hour. But anywhere from an hour and a half to an hour you'll have left during you're not leaving early, if that's what's going on in your mind. There's a part 2, so I decided to do part 2. So this is part 1. Part 2, I thought, how can I do a low stress assessment? Because that's what a test is an assessment to see if you understand the material. How can I do a low stress assessment? Bless you. Oh, it's a cough, sorry. So how can I do a low stress assessment? Well, that gives me the information I need, but then keeps you calm, right? And it's the last day before winter break. You have some other work you have to do over winter break. It's not going to be that much by any means. It'll just be another chapter, some more assignments, just like the stuff you've been doing, right? So it's not like the end of the world. It's not like, oh my God, I have to spend my whole winter break in the library. Nobody's asking you to do that. But in fact, you could probably do all the work that I'm going to assign you for your winter break. You could probably do it in a day. And you have 3 weeks off. You're not going to do it because it would take me a long day. You're probably going to break it up. And you'd be smart to do one assignment a day, because then it's just easy, and then it keeps your brain thinking about it for all 3 weeks, and then, you come back refreshed, but still not losing any of that content, which is the point of the package, right? Also, it's reviewing some of the stuff you should have learned in middle school. So, and I know a lot of you have, but some of you haven't, so you need refreshers, right? Some of you have learned it, but then forgot it, which happens to all of us. So the winter break package, that's what it's designed to do, all right? It's not designed to make your winter break a living nightmare. Just a little extra work to keep your mind fresh. That's it. But it's still worth a lot of points, still important. We're going to move on. So what I decided was you're going to make a poster. It's going to be a poster creation. I'm going to grade it right there on the spot. And it'll be something like draw a cell or cells on a poster, so you can draw an animal cell, a bacterial cell, whatever you want, right? Draw a group of cells and make sure you diagram out photosynthesis, respiration, chloroplast, mitochondria. The poster creation will be teamwork, so there'll be a team working on it. And also, you'll have resources available to you. What that means is that you're going to be able to use a Chromebook or, to make the poster. As long as everyone's done with, as soon as you're done with the test, as soon as you're done with part 1, you're going to move on to part 2. If you work for 2 hours on part 1, that's all you have to do. If you're going to sit there and work on your multiple choice, that's all you want to do for 2 hours, and it's only 30 questions, and you need full 2 hours to work on those 30 questions, you can have it. You don't have to do part 2. But if you're done early, you do part 2. You'll get credit for completing it, and you'll be graded on how accurate it is. And did you meet the benchmarks? Did you meet what the rubric says you have to have? And you'll receive a rubric on test day that'll show you the question, the rubric, and then you'll have your devices, and you as a team are going to create a poster that meets those things in an hour. It'll be an hour-long assignment. You could probably do it. I'll make sure to design it so you can do it in a half an hour, but you're all going to have to bring colored pencils, and you know what I'm saying? So you have to be ready. I'll have paper for you. I'll have poster paper for you. You're going to work on the lab benches. You can work on the floor. You guys can sit there and make it as nice as you can. The nicer it is, the higher the grade, as long as you meet each of the benchmarks. Does that make sense? Does that sound like a reduced stress exam experience?
Yeah.
Okay, yeah.
Is it more or less extra credit, or can you fail it?
Honestly, the poster creation, where you're going to fail it, yes, you can fail it, is not extra credit. But it is a way to boost part 1 up, right? It's part of your final. So I don't know what percentage, but it'll probably be something like 70% the multiple choice of the final, if this is your final grade, right? Mm-hmm. Then of that, 70% is going to be part 1, and then the rest is going to be part 2,
Okay.
right? something like that. I'm not telling you 70, I haven't decided yet what the percent breakdown is. But now, like I said, if you feel like you need the full 2 hours to work on question 30, then you have 100% part one. I have no problem with you just focusing on part one. Like I said, I will test all the concepts in first semester with just the 30 questions. That's all I need to know from you on whether you understand the material or not. You might ask, how do you ask 30 questions that cover all that material? Well, remember, if we're talking about photosynthesis, then we're talking about proteins and lipids that are making carbohydrates using light energy. Guess what that covers? That covers a whole month and a half of the chemistry of life, right? So there's really no need for me to test you on covalent ionic bonding. All that first month and a half to 2 months was just about getting you ready to be able to handle photosynthesis and respiration. That's the whole point. Now, those people that did kind of blew off the first couple months have a real hard time now trying to deal with photosynthesis and respiration, really understand what's going on. The final's still going to be worth a lot. It's going to be the biggest test of the semester. And remember, your semester grade is the grade that defines whether you pass this class or not. So you can have an A first quarter, okay? And second quarter, if you have an F, you fail second quarter, it'll be averaged out. Depending how high the A is, how low the F is. I mean, you could get the F or the D, and that's what goes on your transcript. So it's the average of the two grades, but remember, this is a weighted average. The second quarter is more important than the first quarter. So it's a matter of final designation of the grade is going to be dependent on how well you do this grade, whatever that grade is, at the end of the semester, is the grade. The first quarter report card is just almost like an advanced interim report, right? So make sure you're catching up, keeping up with your work. Make sure and by the way, the winter package, remember, you want to start off the second semester. You're starting all new second semester, right? Start off the second semester getting the work done on time so we don't keep falling behind and running to catch up and falling behind and running to catch up. And then you get exhausted and frustrated and you're not doing as well as you'd like and you're not doing well in the grades. The falling behind and catching up, that's what stresses you out. That's most of the time, that's what stresses you out. It's not the work itself, it's that you blew off the work the first 4 days of the week and you're trying to do everything the day before the exam, or worse, the day after the exam, right? And then you fail the exam, you're turning the work late, you got more work on top of that, you feel like you're getting too much work, but it's not that you're getting too much work, it's that you're not keeping up with the work you do have. Does that make sense? So that's the secret to life. Keep up with the work you have. Not that you have to work all the time, but spread it out so you don't feel like you're overwhelmed. You don't want to keep on taking new responsibilities on, though. Oh my gosh, I have extra time. I guess I'll go ahead and join a club. Or, you know what I'm saying? Don't do that. Just make sure you're taking care of your business. All right. So that's the test. For the final, we're talking about the final. And now, for next week's test, next week, it should have been today, right? But we moved it back because of all the craziness that's happening this week. And next week's test is going to be on what? You tell me. That's right. DNA replication. That's section 2 of your homework. Hopefully you've already started reading it. DNA replication. And of course it'll be something called transcription. And another thing called translation. Today I'm going to kind of outline it for you, okay? And DNA replication, transcription, translation, also mutation. Remember, all 4 of these topics are on your end of course exam. And ultimately, okay. Fine. Transcription, translation, DNA replication, mutations, all are going to be on next week's exam. And I'm thinking really that it's gonna have to be on Thursday, right? To make sure we covered it, I wanna spend Wednesday. I wanna spend Friday certainly asking if you have any questions, Friday and Monday asking if you have any questions. I wanna spend some, that kind of time on it, because this is really stuff that's gonna be on there. I'm gonna, obviously, who's done the homework? Who started to do the homework? All right, some people have, some people have. Section 1, Section 2, you should be done finishing Section 2 by now and starting on Section 3. This is going to be due on Wednesday, the day before the test. That makes sense, not the day after. There are no makeups anymore, no turning in things late. All this stuff, see all this? This is every time I let you turn things in late, right? This is what happens. My work backs up. Like I tell you, don't let your work back up. This is what happens. Your work backs up, which makes doubles and triples the time it takes for me to get stuff in the grade book. Does that make sense? So that's why I have that rule that no makeups, no retakes after a week before the finals week. And finals week is in 2 weeks, so next week, no makeups, no retakes. However, because I missed a day yesterday because of the field trip, because during advisory you could have retaken any exam you wanted, because people didn't have that day on Thursday, on Thursday when we had the field trip, I'm going to give you Monday during advisory if you want to retake a test. I warn you, they're going to be essay tests. So that's what the kids had to come in. I say essay tests, I mean I'm going to give you a question, I'm going to ask you to describe it, explain it, diagram it, that kind of thing. Okay? All right, so that takes care of the test. We covered 2 big issues, the final and the test. I hope you feel better about that. The blueprint will be in your mail, in your grades by tomorrow. Now let's talk about what's funny. What? Oh, mailbox. Yeah, well, mailbox. AOL used to call them mailboxes. Gmail, it's still called an inbox, right? And your Gmail, right? Inbox, outbox, drafts. All right, so let's go on and start talking about DNA replication. So, what's the first section? Those of you that did the homework, what was the first section about? Before we go on, I want to show you what Google has on their search bar this morning. What is it? What? Photosynthesis. On the search bar this morning. It's Jan Ingenhaus. I think he was Swedish, it might have been German. Yeah, he's not alive, he's dead. But he was born 287 years ago, and he's the one that first described photosynthesis. Huh? Oh, today's Sinead O'Connor's birthday? Oh. She sent her Instagram, or what is that called, a tweet letting her know that her and Jan share a birthday. And that's the guy that developed photosynthesis. I bet she'd appreciate that. All right, so let's move on. Let's talk about DNA replication. What's the first section 1 of chapter 12 about? Genetic material. What is that genetic material? Is one, yeah. What other genetic material did they talk about in RNA? That's right. You guys did an assignment. If you didn't get that assignment, it's still up here. That assignment is due. I expect you guys able to, I expect you guys doing it, right? I expect it done. I can promise you it's going to be on your test next week.
The assignment from yesterday.
The assignment from yesterday, that's right. I understand that some people had trouble with the third link. I know the link works, but I'll work on it and send you the link. If there's a problem, I'll fix it and send you the link. I know the link works is actually change the link to what it is. So I know some of us went to anatomy camp. By the way, anatomy camp went very well. It was very interesting. I know you have a lot of homework to do as well. So if you have a choice whether to do the homework or do this, do the homework. The homework is more important because it's directly related to the test, especially those practice questions. Okay? So, but I think this, what did you guys think of the work that you were able to do in class? Was it something that was related to the chapter work? It's useful? Okay, so I think it's useful to do it then, if your colleagues think so. So we have genetic material, and there's DNA and RNA, and kind of what do you need to know about DNA and RNA? You already know all the experiments that helped us understand that DNA was the genetic material. We have DNA and RNA. What is the difference between the two things? There's two major differences. One is DNA is double helix. That's kind of a takeaway from the chapter, section 1 you should do, right? Double helix. RNA tends to be single-stranded. Single-stranded. DNA is double-stranded. Double-stranded and in a helix. Helical pattern, meaning it's winding. They wind around each other. There are two molecules. DNA, when we talk about double-stranded, there are two strands, two molecules. They're being held together by hydrogen bonds, right? These hydrogen bonds hold the strands together. Now it's important that you understand they're hydrogen bonds because hydrogen bonds means that you don't have to chemically break them up, right? You don't have to change a covalent bond, which means you don't have to have an exchange of electrons in order to separate the two, right? If these were covalently bonded, you'd have to actually have a chemical reaction change them rather and exchange electrons being exchanged in order to have a separation of the two strands. Yes.
Isn't a hydrogen bond because it's a weak one or something?
That's right, because they're weak and it's intermolecular. Good. Because these are weak and they're intermolecular, you can separate them a lot more easily. So the conditions that you put this under that separates these two strands doesn't separate the carbons in the sugar phosphate backbone, right? So you can separate these two without breaking up the strands. So what do I mean by that? Who here has had their hair braided or has braided hair or made anything like that? So do you have to break the hair to unbraid it? No, you can just untwist it. How hard? You can pull pretty hard on hair and not break it, right? But interestingly, so the things that are holding that hair together as hair are much stronger than the twisting that's holding it together as a braid. Do you agree? Well, it's the same thing with DNA. The hydrogen bonds that hold the two strands together are weaker than the chemical bonds, the covalent bonds that are holding the sugar phosphates together. So we can unwrap it without breaking it, without breaking the molecule up. Does that make sense? So just like you can unwrap hair without breaking it, you can unwrap DNA without breaking it. Yeah, question?
You said the hydrogen bonds are weaker than the chemical bonds?
Then the covalent bonds. Covalent bonds. I did say chemical, I apologize. I meant covalent bonds. Are we good with that? Okay. So the letters, the ATGC, remember they're what? They're the nucleotides. They're here in the center, right? They're your nucleotides. They're the things that are doing the hydrogen bonding, right? And they're in the center. There's one on each side. That's where you get like your A hydrogen bonded to your T, right? And you have your G hydrogen bonded to your C. Notice there's 2 hydrogen bonds. Here's 3 hydrogen bonds, right? So that when we say ATGC, what we mean is not this. What we mean with the sequence A, T, G, C, A, T, G, C, etc., right? These are covalently bonded together. Do you see that? They're bonded together with sugar phosphate backbones. There's another strand that's T, A, C, G, T, A, C, G. And these are also covalently bonded to one another. So they're strongly bonded, like your hair is strongly connected to itself, right? But then when we braid it, what they do is we make hydrogen bonds between the A and the T. I'm just going to put one line because I just want to show you that they're hydrogen bonded. So these red bonds are hydrogen bonds. All right. So even though these are covalently bonded to each other here, they're hydrogen bonded between the strands. Does that make sense? So you got this DNA. Does that make sense to everybody?
Yeah.
So if there's, and this is like one of the last times we're gonna have to talk about this before the test. So make sure you ask a question. And of course, you don't have a lot of questions if you haven't read the chapter yet and you haven't done your homework. You've had a week to do it now. You really should be pretty far along on your homework. I know it's stressful, but it should have been done. All right. So now, DNA, you have a nucleus, and it's in a cell, and even the biggest cell is pretty small, right? Do you agree it says cells are pretty small? We call them microscopic, right? Do you agree? They're small. This DNA is inside the nucleus, and that's all the DNA you have. I'm going to go ahead and use humans as an example, all right? Humans have 23, and you need to know this, because we're going to talk about this next quarter too when we get into genetics. 23 pairs of chromosomes. 23 pairs. When the DNA is, a chromosome is when a DNA is all wound up. Like, I'll be honest, hold on a second. They're all wound up like this. I love dreadlocks because they're really a great example of what I'm talking about. It's hair, taking hair that normally is kind of like spread out, you can wrap it around and make it in a nice tight wrap, right? Well, that's what happens with DNA. DNA is all wound up around histones, these proteins called histones. Yes, you should know what those things are. They're called histones. They were in that packet yesterday, right? They're called histones, and histones are just proteins. All you have to know about histones is that they are the proteins that the DNA is wrapped around. That's it. They help regulate gene expression. We'll talk about that later, but right now, what you need to know, I keep telling you, I'm going to point out what you need to know, right? So you need to know that proteins are histones, called histones, are what the DNA is wrapped around. I know I don't know if anybody still uses curling irons or they still use hair rollers, right? Has everybody seen someone use a hair roller? My grandmother, okay. So a hair roller is like a histone. Okay, so if you take that, your hair is the DNA, the hair roller is a histone. Okay, so that's all wrapped up in a nice tight braid, or if you want to call it dreadlock, whatever. This is one chromosome, one copy of the chromosome, and this is when this only shows up during a phase of the cell cycle known as synthesis. And that synthesis phase is when you're doing DNA replication. What do we call it? What do we call DNA replication? What does that mean, DNA replication? What does that mean? You're making a what? A copy. When you made a copy of the DNA. So this is two strands, we just talked about that, right? Two strands all wrapped together. This is one copy, and this is the second copy. Do we get that? Those are copies. We have one of those. So after DNA synthesis, this particular one came from, let's say it came from dad. This chromosome came from your father. After DNA synthesis, you have two copies of your father's chromosome in you, in that cell. Are we clear? The chromosome that came from mom, there's a chromosome that came from mom, just using a different color, also wrapped up the same genes. Both chromosomes have the same genes. They have different, what we call alleles, different versions of the genes. Also have histones. They have histones as well. That's how you can wrap. You can't wrap it up otherwise. These DNA molecules, as tiny as they are, we're talking at the molecular level, they are 3 meters long. And we're wrapping them up so tight that they fit inside a tiny little nucleus inside a cell. Imagine how tight you have to wrap that up. But in any case, this is mom's chromosome, and there's still chromosome number, there's 23 chromosomes, pick a number. 4. They're both chromosome number 4, but you have 2 copies. Why do you have 2 copies at the end of DNA replication? Because it's making a copy. That's why you're doing DNA replication, because you're making a copy. You need a copy of exactly the same material in order to have two cells to have the same DNA, right? Every cell in your body ideally has the same DNA. By the way, write that down. Every cell in your body, except for some interesting exceptions, every cell in your body has the same DNA. That means there's some big implications on that, really big implications on that. But your eye cell has the same DNA as your brain cell. This is a question that trips kids up all the time. Your eye cell has the same DNA as your brain cells, has the same DNA as your heart cells. Same exact sequence of nucleotides. Yeah.
So you know how when females get their tube side, they mean that they come from a copy.
No. That's a good question. I guess that's it's an interesting question. Tubes tied, what you're talking about is stopping sex from happening. It's stopping the sperm cell from reaching sex in biology means the combination of DNA. So you're stopping the male DNA from reaching the female DNA. So we stop the sperm cells from reaching the egg. How does a sperm cell get to the egg? Through a tube. It's called the fallopian tube. So if you are a fertile woman, then you have a fallopian tube where sperm can swim through it to reach the egg. Does that make sense? So if we tie a knot in that, literally that's what they do. They tie a knot in it. Not literally, but in a process that they call tying a knot in it. If you tie a knot in that tube, the sperm can't get through.
Does it get blocked?
It dies. It gets reabsorbed into the woman. What?
I thought it get blocked.
I can't hear you.
I thought it would get blocked or something.
Yeah, it gets blocked. By putting a knot in it, you're closing the tube up. Your body sometimes unties it and finds ways of working around it. So some women that get their tubes tied, and men can also get their tubes tied because we have tubes that go from our testicles to our to the other male part. I don't want to say the word because I'll get giggling. But go from the testicles where the sperm are made. I'm already getting giggling and I didn't even say it. From the testicles to the other place, and those little tubes can be tied as well. So you can get males or females can get their tubes tied. It basically makes it's a permanent contraceptive, ideally, without using chemicals. But this is at the cellular level.
You can't get one cell?
Yeah, you can, but I'm just saying it's basically permanent, right? So we're talking about cells, guys, DNA being copied. Don't use the cell phone in class or I'm going to take it away. I've taken them away all day long. I think I'm going to start holding them till the end of the day so you'll go to your next period without it. So please put them away and pay attention to what I'm saying. The DNA gets copied, and the only reason you're copying is so that you can split the DNA in half of the total into each of the two new cells that you're trying to make, right? So when you're talking about cell division, you have to think about it. Each cell has to have the same DNA, the same amount, but also the same quality. Otherwise, your cells would not have the same information, right? You could not. So you need to have that. You need to understand that fundamentally. That's called DNA replication. There's 23 pairs. There's 23 chromosomes, numbers 1 through 23, and there's 2 pairs. There's a pair of each. So what that means is you got one of the 23 from mom, and you got another chromosome, same number, same genes, from dad. They're different versions, so you get the gene for hair color, but maybe dad's hair color's gene is brown, and mom's hair color gene is blonde or red, and together you get whatever color hair is a result of that. Does that make sense? So that's why when you did those Punnett squares in middle school, those of you that did it, you used to have little a, or big A. This was a gene on one chromosome, right? And this was a gene on another chromosome. This was mom's gene, and this was dad's gene. This is on the chromosome mom gave you, this is on the chromosome dad gave you. So if you had big A, mom gave you a big A, dad gave you a little a. If you have big A, both parents gave you the same version of the gene. But this would be a chromosome. This is a gene, an area on the DNA that codes for a protein. Write that down. That's an important definition. A gene is an area on the chromosome that codes for a protein. An area on the chromosome that codes for a protein. That is a gene. Codes for a protein. And we'll talk about that. There's details that I don't want to get into right now. But go ahead. That's right. If you want to see something before you leave, there's a poster up here that looks at the chromosomes. All right, I'll be taking a picture of it and sending it to you because and we'll be using it in class as well. But go ahead and take a look at it on your way out the door if you want to spend a couple minutes looking at it. We'll stop the class a little early. And you see Down syndrome's the picture of those genes up there, of those chromosomes up there. So like I said, if you're going to divide a cell and your cell needs 23 pairs of chromosomes, you need 23 pairs going into one cell and 23 pairs going into the other. That's why you have to go through the synthesis phase of the cell cycle that copies that DNA. So you end up with how many at the end of DNA synthesis? 46 pairs. So how many total chromosomes? At the end, in the one cell, the big old giant cell without a nucleus that has all these chromosomes that you copied. See how they have we call them chromatids. You'll go learn it. How many? 92. That's right. Now you have 92 in a cell in a human cell that's going through cell division. Every species has a different number of chromosomes with different genes on each chromosome. So that's one of the definitions of species. That's how come you can't reproduce with a chimpanzee, because they have different chromosomes with different genes on them. If you reproduce if we take a cell from you and try to combine it with a chimpanzee, even though our DNA is very similar, where the genes are located and how many chromosomes they have is different. So your babies will either have too many or too few, and so the difference is going to be so bad it won't work. It's like making a car without an engine, or making a car with 5 engines in it. It doesn't work, right? You're gonna have the car won't work. So that's what happens with species when they try to have intercourse with each other and they can't. It's because one major reason is chromosomal differences. Yeah.
If your parents give you the same genes, don't that mean that your parents are related?
Yeah, your parents are related to you. They defined you. Yeah. No, there is no stupid question. If you don't ask it in class, you're going to get it wrong on a test. But yeah, your parents are related to you. It's not exactly the same genes because you have that thing called mutations, right? So you have there'll be some differences. And they don't have the same combination of genes. And that's because of something called meiosis. So there's 2 sources of genetic variation. Do you understand this word variation? Variation means differences.
Like a variety of chips.
Yeah, like a variety of chips, right? So genetic variation, means all the different genes in a population. So you have a lot of genetic variations in this classroom. There's a lot of genetic variation in your own family. Even though the source of all you and your brothers and sisters' DNA came from mom and dad, assuming you had the same parents, even though they came from both your parents from the same source of the genes, you recombine them, you remix them up. So maybe dad has 2 dominant skin color genes and mom has 2 recessive. Skin colors are bad. It's bad to do humans at all because we're so complicated. But it's not just humans. Life is so complicated. But you can pick a trait. I don't care what that trait is. You have, let's say, simple genetics, right? Dominant or recessive. You can have dad has 2 dominant versions. Mom has 2 recessive versions. You would think that in the end, and it's true, you would get one version. You know what? I'm getting ahead of myself. If I start talking about this, we're going to end up spending the rest of the period talking about that instead of what we need to talk about. So I'm going to stop right there. We'll get to that when we get into genetics. Yeah.
But I don't know
But I had the question about like, okay, so like me and my brother, if me and my brother have the same mom, like why don't we come out with this?
Right, that is exactly what I was just trying to get at.
Okay.
But it's a good question, it's an excellent question, but I just don't want to talk about it right now. We will get to that. But I literally will I won't even get to the answer. All right. But I'll have to explain a lot to you. So we have these chromosomes. This only shows up, this thing that you see here, I'm sure you guys have seen people draw before. They keep drawing the same kind of structures, right? We call this a chromosome. Let me tell you that this is only present during DNA, right after DNA synthesis, right during a phase this shows up in a specific phase of mitosis. Most of the time, the DNA is all like this, all unwound. Maybe there's some pieces that are wound up here, and then there's more. It's sort of like us as human beings. Most of the time, our hair's not done, right? But when we're going out in the public, we get our hair, we make sure we comb our hair and fix it up. You're at home, I bet you, if your parents recorded every day of your life, like 90% of the days you're at home, your hair's just a mess. Especially on a Saturday, you're relaxing, playing video games, you get up only to pee and eat chips, and that's it. And your hair's a complete mess. You're lucky if you brush. Oh, that was me. All right. So anyways, I guess I like playing video games. I think it's fun. I forget about everything except eating and peeing. But anyways, it's funny. No? All right. So anyways, the point is that your hair is usually a mess unless you're going out. Well, in DNA, your DNA is usually unwound. We call this unwound form chromatin. This is a chromosome. All right. Interestingly, when your chromosome is in this form, we call each of these a chromatid. There's 2 chromatids per every chromosome after DNA replication. Now here comes the hard part. It's hard because people don't pay attention, and then they get on the test, and they get caught, and then they say, whatever. When they're together, we call them chromatids. When they get separated, and they are going to get separated, remember this is what we said, this was chromosome 4, right? So this is chromosome 4 from mom. Oh, let's call it from dad because I ran out of room. So this is chromosome 4 from dad, okay? There's another chromosome 4 from mom. They're going to get separated, one into each cell. When that separation happens, this one's going to have one of these chromosome 4s. These are copies. Chromatids are copies. And we know now how we know that they're copies, right? We went through the experiment. So this is one chromosome that's going to go into cell number 1. Let me not number one. This is going to come into the left cell over here. Into this, it's going to turn into that, and then the other chromosome is going to go what? The other chromatid is going to go into the cell over here, right? So you have 2 cells. Is that understood? They're going to split into 2 cells. The chromatids are going to move into the different sides of the cell. Then the cell's going to pinch off, and you're going to get 2 sets. One of the copies going into one cell, the other of the copies going to the other. But as soon as these chromatids, these copies separate, as soon as they separate, we don't call them chromatids anymore. What do you think we call them? I like that. I wish. We call them chromosomes again. It's still chromosome number 4, except there's 2 of them. And really, this leads to the most confusing part of biology for a lot of students, for this section of biology for a lot of students. This leads to the biggest confusing part. Because, we're going to say that humans have 23 pairs of chromosomes, right? There's 21 autosomal, then there's an X or an X and a Y, right? So it's 23 pairs, right, of chromosomes. the 22nd, I mean, 20, there's 22 plus 1 is 23, right? The sex chromosomes are different, right? So you can all, they're all listed up there on the poster. So the confusing part is that when they're together, the whole thing is called a chromosome. When they're separated, each one is a chromosome. That's really gets to be a bear for some people. They get it that, I'll ask them what is this, and someone will call it a chromatid. I'll ask what's this, someone will call it a chromosome, and they got it wrong because they didn't get this right away. So get this in your head right away. Yeah? Huh? I don't know what she said. So this is a chromosome, and when they separate, each one becomes a chromosome. When they're together, it's called a chromatid. When they're separated, all unwound in the cell, we call it chromatin. Now, do you see why I don't like these words that are very similar to one another? It makes it hard for someone who's learning the subject to really learn the difference, but you need to know the difference. You have to, or you're not going to get the questions right on the test. People ask you, why am I getting the questions wrong on the test? And I'll tell you, you don't know your vocabulary. You have to know you just have to know it. You have to learn it. Yeah.
Those squiggly lines, those are chromatin.
Chromatin. It is chromatin. It's still the same molecule, right? The DNA double helix, but it's unwound. It's hair that's unwound. It's DNA that's unwound.
And then those are chromatin.
Chromatin, T-I-N. Dude, my brother is an idiot. I'm going to have to yell at him. And I don't know if I'm recording.
What is your brother?
Oh.
Wait, what do you do?
Anyway, sorry. Let's move on now that my personal crisis is over. So, 23 pairs. And chromatin is unwound. You have to unwind DNA in order to read the sequence. You can't read it unless you unwind it, unless you open it up. I'll show you what I mean in a minute. Is that clear to everybody? So that's why you have chromatin in the normal functioning cell. This, you only wind it up when just like your stuff. You only pack it, you only put it in boxes and organize it when you're doing what? Moving. When you're moving. You only pack when you're moving, right? So the chromosome, you only see this packed form of DNA when the cells are about to divide. Is that clear? As soon as they divide, they unwind. And then you start doing your job. And it stays like that until it's ready to divide again. All right, so, I think we actually, we're probably going to be short like a couple ideas before we're done with DNA replication. So in order to copy the DNA, you have to unwind it. It has to unwind to two sides. Remember, it's a double helix, right? Not only do you have to unwind it from the histones, so it's all wrapped around a what? A histone. And there's a whole bunch of histones. And they're in a chain. You see that? So you have to unwind it from the histone. You also have to unwind the DNA itself to expose the two sides. Because if you're going to make a copy of each side, you have to be able to read, right, the sequence. So when we say ATGC, we mean there's A here, and there's T here, and there's G here. And over here on this side, you have T, and here you have A, and here you have C, and it keeps going, obviously, right? Right? So we have this kind of sequence going on here. They're unwind. You have to open it up so that the machine that's going to read it, can read this what's here and make a copy of it over here. So you're going to start making copies of that. And you're going to start making a copy. It's actually not a copy. It's what we call a complement. So when the machine reads a T, what's it going to put on the other one? An A. And when the machine reads I say it's a machine, it's a molecular machine, it's not a human-made machine, right? It reads an A, it's going to put a T. It reads a C, it's going to read a. And the same thing over here. Here it reads an A, it's going to put a T. Reads a T, it's going to put an A. Reads a G, it's going to put a C, etc. Right? In the end, you see you're going to have the same molecule, right? You're going to have a molecule, a copy of this. You're going to have a copy of this thing here. It's just they're going to have this side's going to have this sequence. This side's going to have that sequence. You see that? And they're going to wind together and form their own DNA. Except that's why this is semi-conservative. One black strand is going to go with the blue. You see that? And this black strand is going to go with this blue. So one new and one old, semi-conservative. And these match up. They're the same. You see they're going to be exact copies, but you'll have two copy versions of them. In the end, you'll end up with those two versions stuck together into something like, and wrapped up around histones that look like this. One version's here, the other version's here. They're both copies. They're both chromosome, in this case, the example we've been using is number 4. They're both chromosome number 4. Are we good with that? That's really one of the major concepts you really need to know. I'm gonna say, please do your homework. We'll continue this on Monday. Get the homework done, especially DNA replication stuff.